This is the final lecture in our Streptococcus lecture, and in this lecture we're going to focus on the laboratory diagnosis. Again, for all of our Streptococcus species, these are gram-positive cocci. For Streptococcus pneumoniae, they tend to be lancet-shaped. They can be in pairs or in chains. For Streptococcus pneumoniae, they tend to always be in pairs, which is why it's called Diplococcus. These are non-spore forming organisms. These are very teeny tiny organisms. So if you have a staph and a strep on the same blood agar plate, the strep colonies on the plate tend to be much tinier than staphylococcus colonies. You always have to look very carefully for any type of hemolysis in your Streptococcus organisms on sheep blood agar. Here's your nice chaining of your Streptococcus organisms. Now here's a mixture of Streptococcus and Staphylococcus in a gram stained specimen. So in this specimen you can see some white blood cells, those pink large cells in the background, and you can see your nice Staphylococcus. They're a little bit bigger organisms right in the center of the slide here, and you can see them clustering together. All around the slide is Streptococcus, which, and to me it looks like a Streptococcus pneumoniae, a Diplococcus that's lancet shaped, and you can actually see a little halo of the capsule around it. So in general, on agar media, the strep organisms tend to be larger than, the Staphylococcus tends to be larger than Streptococcus. Streptococcus is usually very pinpoint, teeny tiny. And also under the microscope, Staphylococcus organisms tend to be a bit larger than the streps. However, that's easy to see when you have staph and strep under the same microscope, but commonly you don't have that, so you wouldn't really be able to tell just by looking under a microscope if you had staph or strep. You have to go on and do that catalase test. So again, it's Streptococcus, so they are all catalase negative, and this is a key test to differentiate the staphs and the streps. Streptococci tend to be a little bit on the fastidious side. They are aerotolerant anaerobes, so they don't like to be in high oxygen levels. They grow best in the presence of CO2, so they are commonly grown in a CO2 incubator. These are incubators that usually have between 5 to 10% CO2 constantly going into the incubator to keep them without quite so much oxygen in there. Streptococci, most of the species require some sort of animal cells, so they will grow best on sheep blood agar plates. Some of these streps, such as Enterococcus, will grow on anything, nutrient agar. They don't need any type of cells to grow, but some of the species won't grow very well on your basic nutrient agar, so that's one reason why clinical labs tend to use sheep blood agar, because even slightly fastidious organisms will grow on it. Its optimal temperature, like most of the pathogens, is 35 to 37 degrees Celsius, and streps will ferment sugars for energy. The common diagnostic tests for the streps, we already mentioned all of these: Bacitracin Susceptibility, PYR Hydrolysis, sodium Hippurate Hydrolysis, the CAMP test, the Bile Esculin test, 6.5% NaCl or saline, Optochin, and Bile Solubility. So we're going to go through each of these tests because as medical laboratory scientists, you need to understand how to differentiate one organism from another organism. So for our bacitracin test, it is a 0.04 unit disc of bacitracin, and the disc has a capital A on it, so it's also called an A disc. So again, group A strep or Streptococcus pyogenes is susceptible to bacitracin, so there will be a zone of inhibition around the A disc, and other streptococci are resistant, so they'll grow right up against the A disc. Bacitracin susceptibility is a presumptive diagnosis for Streptococcus pyogenes. These strep pyogenes or group A strep are highly susceptible to bacitracin, so there will be a zone of inhibition around the bacitracin disc. The bacitracin discs contain between 0.02 and 0.04 units of bacitracin, and 99% of group A streps are susceptible to the bacitracin disc. Here's a typical bacitracin sensitivity to Streptococcus groups A, B, C, and D. You can see that nice zone of inhibition around the group A Streptococcus. There's another type of susceptibility test called SXT, or the trimethoprim sulfamethoxazole. This is an antibiotic disc, and you can drop it on your lawn of bacteria and see if the organism can grow in the presence of trimethoprim sulfamethoxazole. So usually trimethoprim sulfamethoxazole antibiotic will inhibit beta hemolytic Streptococcus groups other than group A and group B. PYR hydrolysis is a very quick and simple test to differentiate the group A and group D streps. It tests for the organism's ability to hydrolyze PYR, which has a big, huge, long name, and I won't attempt to say it. The hydrolysis is indicated by the development of a red color, and we're going to show this on the next slide. So all group A and Enterococcus are PYR positive. So here you can see the PYR positive on the right, where you have the disc that contains PYR. You put your organism on this disc, you drop your PYR, moisten the PYR disc, and if it turns a pink to red color, it would be indicative of a positive reaction. And again, group A streps and Enterococcus species are positive for PYR. Hippurate hydrolysis is not a common test done anymore, but it is a test that can differentiate the group B Streptococci from other beta hemolytic Streptococcus species. So the group B Streptococcus or Streptococcus agalactiae will hydrolyze sodium hippurate. Again, diagnostic for group B, tests for the production of the enzyme hippurase, and the detection is benzoate. The CAMP test we already discussed is diagnostic for group B strep. The CAMP factor enhances the hemolysis of the staphylococcal hemolysin in Staphylococcus aureus. So you get that increased zone of hemolysis that is in the shape of an arrowhead at the junction of your Staphylococcus aureus line and your Streptococcus agalactiae line. And here is the CAMP test, and CAMP is nomenclature is for the group of individuals that discovered the test, and they are Christie, Atkins, Munch, and Peterson. So again, it detects that enhanced hemolysis with that nice arrow shape. Here's more CAMP tests. On the top left is CAMP positive, and on the top bottom, CAMP negative. Bile Esculin hydrolysis is another test that can be done, and it shows the ability of an organism to grow in 40% bile and also to hydrolyze esculin that's in the media. And group D organisms are positive for bile esculin hydrolysis. Another test to differentiate those group D Streptococci species is growth in 6.5% NaCl. So the group D streps can grow in 6.5% NaCl, but Enterococcus cannot grow in 6.5% NaCl. So both group D strep and Enterococcus are positive for bile esculin hydrolysis. You then need to differentiate those two by doing the 6.5% NaCl test. We already talked about the bile esculin hydrolysis test. And now we're going to move to bile esculin hydrolysis. So you have media that will contain the dihydroxycormorin complex, and if your organism can hydrolyze bile esculin, you're going to get a dark black color in the media. So that's on the right hand side, you see that dark brownish black color is positive, and left is an uninoculated negative. So you could see the difference in the color reaction. 6.5% NaCl or saline will allow you to differentiate your Enterococcus from your group D strep. So you have your broth that has 6.5% NaCl in it. You must inoculate 2 broths. You have to have first you have to have a control broth that doesn't have the sole. This is going to let you know that your organism will actually grow without sole. And then you need your tube with the sole. And if the organism grows in the tube without sole and the tube with sole, you know it's Enterococcus. If the organism grows in the tube without salt, but it doesn't grow in the tube with the 6.5% saline, then you know it's probably a group D Streptococcus and not an Enterococcus. If it does not grow in the tube without salt, nor does it grow in the tube with salt, you need to start over because your organism is actually not viable. Your control didn't work. So in the clinical laboratory, everything usually has a control. And if your control does not work, you must do the test over. We talked about the susceptibility disc reactions, such as the Optochin disc, the Bacitracin disc. So Optochin is the P disc, and it's going to differentiate your strep pneumoniae from your other alpha hemolytic streptococci, such as your viridins group streptococci. Another test is the Bile Solubility test. Streptococcus pneumoniae can lyse in a suspension of sodium deoxycholate. So the viridin streps will not. So in this test, you have your organism growing on a plate. If you drop sodium deoxycholate on it, the colony will just disappear. And that's indicative of being bile soluble. The Optochin disc contains ethyl hydrocuprion HCl. Streptococcus pneumoniae is inhibited by very low concentrations of Optochin. So you get a zone of inhibition around the disc if it's 14 millimeter in size or greater. This would be indicative of Streptococcus pneumoniae. And here, Streptococcus pneumoniae is on the left. It has a nice large zone of inhibition that's greater than 14 millimeters. And on the right, you have an organism, an alpha hemolytic organism that grows right up against the disc, and this would be a viridins streptococcus. We talked about bile solubility and how Streptococcus pneumoniae will lyse in the presence of bile salts. In this test, there are tube tests or plate tests. tests. In the plate tests, I already mentioned you drop the sodium deoxycholate and the colony disappears. If you have your organism growing in broth and you drop some sodium deoxycholate or other bile salt in it, it will clear the broth. So here we have the broth test. You have Streptococcus pneumoniae with the bile, and you can see it's clear, so that's indicative of a positive test. You always have to have your control tube that doesn't have the bile to prove that your organism was actually viable and able to grow without the bile. And on the right, both the viridins without the bile and with the bile are cloudy, so you didn't get lysis, and that would be indicative of a negative bile solubility test. There are many different types of serological based tests as well. You can test for streptolysin O, you can do DNase tests, you can do a quelling reaction which tests for the swelling of the capsule of Streptococcus pneumoniae, and you can also type the Streptococcus pneumoniae based on the capsular antigens. There are all sorts of agglutination tests. Agglutination is one of the ways to look for your Lancefield antigens. Here's a nice flow chart for your gram-positive cocci. So we showed a flow chart at the end of the Staphylococcus lecture for the catalase positive staphs, and here there on the left hand side you can see the Staphylococcus. This one is not quite as extensive, but it still gives you the idea. You have a patient sample, you have a gram-positive cocci, you do your catalase test. If it's catalase positive, it's likely a staph. If it's catalase negative, you're probably looking at a Staphylococcus species or an Enterococcus species. The first thing you want to do is look at hemolysis, and then based on the hemolytic reaction, you're going to think of certain organisms and certain tests. So you need to know exactly what these organisms and what these tests are to differentiate between the various Streptococcus species. And here's a much more extensive flow chart for the Streptococcus organisms, and I would suggest that this flow chart is one you put into your memory banks. You will need it for your midterm exam. You will need it for your final exam. You will need it when you walk into the clinical laboratories for your clinical microbiology practicum course. You are expected to know these test reactions when you walk in the door, and you will also need it for your Board of Registry examinations. So the best thing you can do is just put this whole flow chart to memory, keep writing it out over and over and over again until you can write that chart in just a minute and just know it by heart. Okay, our case study. We have a 9-year-old boy that complains of a fever and a sore throat. When he's examined, his pharynx is red, his tonsils are swollen, his neck or cervical lymph nodes are swollen, and a throat culture is taken. So what would you want to think about for this patient? You want to look at the clinical symptoms. Okay, this patient has a sore throat. Almost all of the streps can cause a sore throat, several of them more commonly than others, but one that you really always want to test for would be the strep pyogenes, which is the cause of strep throat or group A strep. So you want to think about how would you grow this organism, this throat culture. You want to streak it out, a streak for isolation like we talked about in one of our first lectures on a blood agar. You want to incubate in the CO2 incubator. You want to look for hemolysis. Streptococcus pyogenes is a beta hemolytic organism. You want to think of the tests that you would need to do in order to differentiate if it is beta hemolytic, differentiate strep group A strep from group B strep, and possibly what other organisms could be causing this infection. So you always want to remember your general characteristics, the hemolytic patterns of all of the streps, what tests you need to do, what types of infections are produced, the various morphologies, and any of the specific tests to identify the species.